Hi, everybody. We are continuing with our special senses for vision part 2. So last thing we talked about for part 1 was describing the third layer of the eye, which was your sensory tunic or your retina, which was made up of photoreceptors or neural layer and the pigmented layer. We also talked about the two types of photoreceptors being your rods and cones. And rods were the ones that are involved in the process of creating images or vision in the dim light, versus when we're talking about cones, cones are responsible for creation of vision in the presence of light with high acuity. We also talked about that when rods and cones create their image or create a signal, the signal gets passed down to the bipolar cells and eventually into the ganglion cells. And that signal, ganglion cells axons will become the optic nerve, and that message is eventually sent to your brain. Now we're going to describe the main concept, which is phototransduction. How does your photoreceptors with the help of bipolar as well as your ganglion cells create your image? Excuse me. So before we talk about that, let's quickly go over the two main chambers and the type of fluids that are found inside your eye, and then we'll come into phototransduction. So there's two types of chambers in your eye. You have the chamber that is located in the back, or segment, sorry, that is located in the back, and that is the much larger segments you have. This is basically what you see in the grayish color. This segment is referred to as the posterior segment, which contains a fluid called the vitreous humor. The type of fluid that you can find here is relatively thick. It's gelatinous, and it is important in terms of making sure that your neural layer and pigmented layer are tightly attached with each other. It also helps with maintaining your intraocular pressure, basically the pressure of the eye. And it is the fluid that is actually present since the embryonic development and basically lasts your whole lifetime. Anterior segment, which is, again, as the name implies, it's the front part of the eye, has two types of chambers. So if this whole thing is identified as your anterior segment, it has this part, which is your anterior chamber, sorry, posterior chamber, I apologize. This is posterior chamber, and this space would be your anterior chamber. And those two chambers together make your anterior segment. Now, the fluid that is found here is aqueous humor, implying that this is more watery. This fluid is created by your ciliary muscles, you see right here. It goes through the posterior chamber, come through the pupil and empties into the anterior chamber and gets picked up by your scleral sinus, and then again recycled back into your bloodstream. So what is the importance of your anterior segment? It is important in terms of providing your cornea with nutrients, which we talked about earlier in the PowerPoint. If you guys don't recall, please go back and review your cornea slide. It is also important in terms of lubrication of the structures like your iris and your lens. So, lens. We also talked about lens in a previous slide. We mentioned that lens is held in place by a tiny string called the suspensory ligaments. And suspensory ligaments are connected to your ciliary body. Now, I didn't really go into details about this last time, but when you are trying to create an image that is an image of an object or a condition that is near you versus something or an object that is far away from you, the shape of your lens needs to be adjusted for you to be able to accomplish that. And this change in the shape of your lens is known as visual accommodation. Now, visual accommodation can be done again in two ways. One thing that you have is described is if your sympathetic nervous system is active, and in this scenario, your lens is thins out or stretches, allowing you to focus on objects far away from you. How does your lens stretch? Here it is how it does that. Your ciliary muscles relaxes, which you see in red, and that tightens or pulls on your suspensory ligaments. So ciliary zona is still the same thing as suspensory ligament for what I want you guys to know. So if your suspensory ligaments are pulling on your lens is stretched, and it allows you to focus on the objects that are far from you. Now, parasympathetic nervous system, in other hand, causes the ciliary body or muscle to contract, which allows the suspensory ligaments to become loosened. And because now there is no pressure on the lens will appear more rounded. This allows your lens to focus on objects that are near you and create, again, focusing and creating an image closer to you versus the far distance. Please do note that this adjustment of the lens are extremely important in terms of your capacity to focus on far or near objects. So a lot of times, if you guys ever notice, is when you are staring at your phone and then you all of a sudden look up, it takes a couple of blinks for your eye to adjust, is because you basically force your eye to stay in this position for a while. So it takes a while for your eye to adjust from this phase to what we depicted here. So a stretching of the lens, it takes a couple of seconds for that adjustment to take place. So it's always a good idea to kind of relax your eyes, or don't stare at your TV, sorry, not your TV screen, your phone screen for too long, because it does cause damage long-term to your eyes. Now, in terms of vision and corrective lenses, we do have problems associated with, or medical conditions associated with the shape of the eyeball. We can have what we identify as myopia, which is nearsightedness. So in this case, your eyeball is stretched out or too long, which implies the focal points for your vision would be in front of your eye, rather than on the actual retinal layer. So notice right here, the lens is focusing the image here versus the image that should be focused all the way at the retina. The opposite of myopia is hyperopia, which is farsightedness. In this kind of scenario, your eyeball is too short, which means that the image focal point is behind the eye. And again, in both scenarios, part of your vision would be blurred. So what does that look like? If you have a normal vision, you have the ability to focus on all of the objects near and far. If you have a myopic vision, your eyeball, again, is stretched, so the vision is formed in front of your eye. So your image that is close to you is clear, but the image that's in the far distance is not. So you identify as nearsighted. When you're hyperopic, your lens is too short. Your image is formed the back of the eye. So in this scenario, you can have a clear far distance vision versus your near distance is blurry. Both of these scenarios described can be fixed by adding a corrective lens that allows the light to focus on the actual portion or the retina. The third condition we have is called astigmatism. Astigmatism is when you have uneven curvature either on your lens or the cornea where the light is passing through, and that causes the vision to be blurry at parts. So again, here is the normal vision, here's a person with astigmatism. This is also another condition that can be treated with corrective lens. Now we're going to get to the last topic we're going to describe, which is phototransduction. It is relatively confusing, so please bear with me. I will try to make it as simple as possible, considering we are doing this online. So first thing I want to talk about is the functional anatomy of the photoreceptors. So photoreceptors is a modified version of a neuron, which means they will still have kind of the axon, the dendrites, the cell body, and the axon terminals. So do keep that in mind, because I think that's part of the concept that people forget to imagine. Now the difference is the receptor part, where the light is actually going to be interacting with your photoreceptors, is a part of the neuron called the, or part of the photoreceptor called the outer segment. Outer segment is connected to the second part called the inner segment, and inner segment continues with the cell body and the inner fibers connecting to the bipolar cells. So here's a picture. Let's take a look at the picture to make sure this makes sense. So if you're looking at your eye, the neural layer or your retina had two layers. The innermost layer, sorry, not the innermost layer, the layer that's connected to the choroid layer, this is your choroid layer in red, was the pigmented layer. So this part is your pigmented layer. And what you see here would be your photoreceptors. The yellow ones are your rods, and the your yellow ones are the cones, and the purple ones are your rods. And if you go farther this way, these would be your bipolar cells that are depicted in red. Now, again, notice you have the cell bodies, and the main difference here is these two parts, what we identify as the outer segments and the inner segments. The outer segments is going to be our focus right here, because these are containing what we identify as discs, where your photopigments are located. So let's take a look at this. Again, don't get overwhelmed. We'll do it one step at a time. So this part, if you look at this, this is a kind of a zoom-in version of the outer, let me go back to this. This is the outer segment of your photoreceptors. So outer segment. Inside these outer segments, you have a specific disc, which are basically these structures you see right here. So they are stacked on top of each other, okay? So outer segments are made up of discs that are stacked on top of each other. Now, what do we have inside these discs? What we have is a light absorbing molecule called retinol. So retinol, again, the goal of these photoreceptors are to be sensitive to light. So therefore, what are they going to do? Absorb the light. When these molecules are, retinol is combined with another specific molecule called opsin, they form a new molecule or a larger molecule called the rhodopsin. Now, rhodopsin, which is a combination of opsin and retinol, is only found in your what? In your rods. If you're talking about opsin, the opsin is not identical in the cones. You will have a specific opsin that is associated with the green cones, one that's associated with the blue cone, one associated with the red cone. And the reason they have different ones because they're sensitive to different wavelength of light. So let's go ahead and take a look at this again. If you look at this structure is found inside where? Inside your discs. The discs are found in the outer segments of your rods or your cones. And these rods and cones are connected to your bipolar cells, and that are connected to ganglion cells. Now, what are the rhodopsin made up of? They're made up of this molecule you see in red, which is your retinol. And the protein structure we see as these folds called opsin. Retinal is found in all what? In all cones and rods. That's never gonna change. What is different is the opsin. So opsin can be specific to rods, but if we're talking about the other molecules, the other cones, which is green, blue, and red, they have their own category of opsin. So they are not present in terms of your rods, but they have their own identical, they have their own specific type. So why is this important? This structure is called a visual pigment. This is the structure that is going to absorb the light, and through its absorption changes its shape, which causes a chain reaction that eventually leads to creation of an image. So I'm going to repeat this one more time. What we're looking at here is photoreceptors. Photoreceptors, specifically the one we're talking about, is rods. Within your rods, you have a specific structure called the outer segment, okay? This outer segment is made up of discs that are stacked on top of each other, okay? These discs are packed with what? With a molecule. If you're talking about rods, they are packed with a molecule called rhodopsin, which is considered a visual pigment. Rhodopsin is a combination of retinal, which is depicted right here, and opsin, which is the larger structure that is holding the retinal. Okay, so let's continue. Phototransduction, which is what I keep talking about in terms of creation of the image, is a process by which the pigment of the visual pigment, which is your rhodopsin in terms of rods, are going to capture your lights, photons of light, and through that create an image in the presence of dim light. Now, when your rhodopsin, which is your pigment, your visual pigment, captures the light, it undergoes changes, and when the environment is dark, it also undergoes changes. So what are these steps in the presence of the light versus the absence of the light for the rhodopsin? Originally, when you're looking at the rhodopsin, you have what we identified as 11 cis retinal position, which is basically no light present, and your retinal, that red molecule we described here, is in a position called 11 cis position. So everything's quiet, nothing is changing. As soon as the light is involved, the retinal is going to absorb the light, and it's going to change from the original shape, which is 11 cis, to a new isomer called the all trans position. When that happens, rhodopsin literally gets separated from your opsin. So retinal gets separated from your opsin. So really what we have is rhodopsin breaking down. Now, eventually light is going to go away, which means your pigment is going to regenerate. All trans retinal goes back to the 11 cis position, and that allows the 11 cis to interact with your opsin again and form a new rhodopsin. So here's a picture of that process. Again, look at this molecule. This as a whole is your rhodopsin. Rhodopsin is made up of what? The retinal at 11 cis position, which is what you see right here. And the purplish structure, let's say, that's your opsin. Together, that's your rhodopsin. As the light is introduced into the environment, your 11 cis retinal, which you have right here, changes to what? All trans retinal. And that causes what? The retinol to be changed and separated from the opsin. So now you don't have rhodopsin anymore because the retinol component and the opsin have been separated from each other. Now, when the light environment is diminished, so you go back to the dark environment, your all trans retinol goes back to what? Goes back to the 11 cis. 11 cis is what you started with. So now it can again interact with your opsin unit and form and reform your rhodopsin. Don't make it more complicated than what it is, guys. Stick to the facts. The rhodopsin is a combination of retinol. Try to say it with me as I'm saying it, guys. It's a good review for you. Rhodopsin is a combination of retinol and opsin. When there is no light present, your retinol is in 11 cis position. As the light gets introduced, you are changing the 11 cis retinol to all trans retinol, which causes this retinol to get separated from your opsin. So literally your rhodopsin breaks apart. And as a result, there is no molecule. So this is extremely important in the process of phototransduction. When the light goes away, your all trans retinol goes back to the 11 cis position, which allows it to interact again with the opsin and form a complete rhodopsin. So why is that important? When there is light present, you have your 11 cis, and at that time, you have a secondary protein called a G protein transducer. When the light hits the rhodopsin and the transformation happens from the 11 cis to all trans, this change causes the transducin. So this change that you go from here to here, sorry guys, my pen is not working again. So this change from 11 cis to all trans allows the protein transducin to interact with your new structure or rhodopsin. This form right here is inactive. When the interaction happens, you form what? You activate it. So again, notice the change in shape. This is an inactive, here's the active. When transducin is active, it goes and binds to another protein called phosphodiesterase or PDE. PDE is active. What is it going to do? It's going to take a new protein called cGMP and break it down to what? GMP. So there is a conversion that is happening here. Why is that important? When cGMP is on its own and it's active, it hasn't been broken down yet. cGMP is interacting with a protein channel that allows the sodium and calcium to come in. Now, when sodium and potassium is coming in, that causes the depolarization, which kind of should make sense to everybody because we know that when the positive charges are coming in to the cell, that causes depolarization. However, when cGMP is broken down by your PDE, then that causes this not to be present anymore, right? So that's the scenario, which means sodium and calcium stays outside because their channel is now closed, and that causes what we identify as hyperpolarization. So review this one more time, guys. When there is no light, so imagine this light is not there. When the light is not here, you're at 11 cis retinol, transducin is inactive, PDE is inactive, which means cGMP is attached to your protein channel, allowing sodium and calcium to come in, and causing depolarization. However, when you have light present, 11-cis retinol becomes all trans retinol, which causes the activations of the transducin. Transducin that is active can now go and bind to your PDE. And PDE is going to take the cGMP and convert it to GMP. That causes the protein channels to close, so no sodium and calcium can enter the cell, and that causes the hyperpolarization of my photoreceptors. So why is that important? Let's talk about this. When there is no light, as we just described, when there is no light, your cGMP gated channels are open, right? cGMP channels are open. Pay attention to these words, which means sodium and calciums are coming in, and that means your cell is depolarized. If it's depolarized, at the axon terminal aspect of it, we can call it the synaptic cleft of it, calcium comes in, which causes the release of these red dots, which are what? Your neurotransmitters. These red dots are your neurotransmitters. Here's the importance of these neurotransmitters. These are inhibitory neurotransmitters, which means when they release and interact with your bipolar cells, what they're going to do is they're going to create inhibitory postsynaptic potential. This neuron basically goes shut down. Says, I'm inhibited. When they're inhibited, they're not going to release any neurotransmitter because basically there is no action potential, right? These neurons are not active. And therefore, the ganglion cell also will not create an excitatory signal. Therefore, there is no what? No action potential going down your optic nerve. Opposite scenario, there is some sort of light. So, reviewing what we talked about and then getting into this. Light causes what? 11-cis to change to what? To all trans, right? Which is activates your, what your? Transducin, which activates your PDE. PDE going to do what? Break cGMP to GMP. GMP causes what? Causes channels for sodium and calcium to close, right? And that causes what? Hyperpolarization. So let's take a look. Here, cGMP gated channels are closed, like we discussed here. That causes what? Hyperpolarization. So if this neuron is hyperpolarized, there is no calcium. So in this scenario, you will not release what? No inhibitory signal, which means your bipolar goes like, I can get excited, right? So they are going to get excited and they release the calcium, which causes the neurotransmitter to be released. The neurotransmitter sends a signal which is excitatory signal, right? Excitatory postsynaptic potential, which creates what? Your action potential in the ganglion cells, and that action potential ends at your optic nerve and eventually your occipital lobe. Make sense of this, guys. Make connections for yourself. It is a difficult concept, but I do expect you guys to be able to say what is happening in each of these steps. Do remember, guys, that what I described for you is only in, what I described to you is happening in rods. So the retinol and opsin is specific in this scenario to your rhodopsin, which is associated with your rods, but retinol is present in all of your cones as well as your rods, and the opsin component is the one that changes. So opsin is specific to your rods, but you have a specific ones for your cones as well. It's not like your cones don't have it. So what is the passage of the electrical impulse through your eye? Again, you're starting with your photoreceptors, and you can write photoreceptors as rods and cones, which passes the message to bipolar cells, which passes it to the ganglion cells. The ganglion cells' axons become what we identified as the optic nerve. Optic nerve, remember the whole concept of contralateral. If these are the eyes, optic nerve is coming out of the back of the eye, but they're gonna crisscross, right? Because we wanna do contralateral concept. So the point where the crisscrossing happens is your optic chiasm or optic chiasma. You continue this as optic tract. So this is your optic nerve. And then you eventually ends up at your occipital lobe. So that's about it. Please, guys, again, go back, pay close attention to the passage of the signals for terms of phototransduction, starting with rhodopsin, changing a rhodopsin from 11-cis to all trans, conversion of that to transducin, or sorry, activation of the transducin, which causes the activation of PDE, which activates which breaks down the cGMP to GMP and allows this process to continue by deactivating your sodium calcium channels, hyperpolarizing your photoreceptors or your rods, which causes excitations of your bipolar and eventual action potential created in your optic or your ganglion cells and optic nerve, ending at the occipital lobe of the brain and creation of the image. I need you guys to be able to, again, recreate this in terms of absence of light, what is taking place versus if you have dim light or very little light or a lot of light. That allows you to create a vision using your photoreceptors. That's about it. We will continue our lecture with our hearing or auditory sense.